It's great to be here. Great to be at Nano HQ, hearing about all these fantastic technologies with the people that developed them. So nice to be able to present to such an expert audience here, and obviously online as well. I obviously going to talk to you about Kv3 channels, as you just heard. I'm going to cover quite a bit of ground actually, and I will define a number of terms which may not be that familiar to you as I go along in the presentation. In terms of an actual outline, I'm going to tell you a little bit about Autifony Therapeutics in a moment, just one slide, but just useful context and something I want to share with you. I'll then tell you about Kv3 channels, obviously the main topic of my talk, and how that relates to, after many years of work, looking at therapeutic options in the clinic for a number of different diseases. I appreciate there are many acronyms here, but again, I'll define these as I go along. And finish by talking about interneuronopathies, which will hopefully become very clear about its relevance as we progress. So, Autifony Therapeutics, we are an ion channel modulator company. No surprise that this is the main expertise within the company that's been developed since this was founded from GSK more than 10 years ago. Just started as a handful of individuals. We're now up to 35 people, and this incorporates in-house biology, particularly electrophysiology, chemistry, clinical expertise, etc. And that's split across a site in the UK in Stevenage and a site in Italy in Padova. And we have Nadia Pilati from the Padova site here today as well. So I say no sort of surprise that we've been working on ion channels for some time. We don't just do it alone, though. We do it with an international group of collaborators and CROs. We can't do everything in-house. We work with the best in the world to progress our programs. We've worked across many of the superfamilies, or the subfamilies, rather, within the superfamily of ion channels. I myself have worked across potassium channels, sodium channels, trip channels, and been involved from programs beginning from idea, just getting nascent programs off the ground, all the way through to taking things to FDA and EMA approval as new medicines. Autifony is proud to be a pioneer, but also the leader in the field of Kv3 ion channel development. We've operated a number of clinical trials now, phase 1, phase 2 trials, developed translational and biomarker approaches, and worked in the rare disease area, as well as opportunities for indications. And something I just want to talk about briefly now, because I won't come back to it in the rest of the presentation, is our early stage pipeline. So as the Kv3 channel programs progress into the clinic, you could say, what next for Autifony? And that's where we think about the next ion channel targets that get us most excited about developing therapeutics. We're not just talking about working on the next nearest neighbor of Kv3, therefore, this is all about what has the highest human target validation, what convinces us that we can make a difference therapeutically. So we end up, for that reason, working on a whole range of different ion channels. Some are ligand-gated, some are actually other voltage-gated channels, but an area of particular expertise, something which Niels has already touched on, we also have an interest in lysosomes, lysosomal biology. We also work on neurovascular channels as well, so we really are diverging our interests as this early stage pipeline progresses. Some of these programs are actually already partnered. We have a collaboration with Boehringer Ingelheim. We also have 2 ion channel programs partnered with Jazz Pharmaceuticals, but we also have some of our own programs still progressing unpartnered at present. So Kv3 channels, then let's put these in context. We have the dendrogram on the left. You can see with the arrow where the Kv3 channel family is. There are 4 members, Kv3.1 to 3.4. They have a single subunit 6TM structure, 6 transmembrane segment structure, and 4 of those subunits come together to form a high voltage activated potassium channel. So they're generally open at more depolarized potentials, far more depolarized than the cell resting membrane potential. And they help to fine-tune action potential firing. And this is predominantly in interneurons, and actually predominantly in a particular class of interneurons as well, parvalbumin positive interneurons, or PVINs. So Kv3 channels can actually essentially drive or increase inhibitory neurotransmission. Now, across the bottom, I have a number of different ion channel medicines there captured. There are no Kv3 medicines currently, so no approved medicines target Kv3 channels, and that's obviously what we're in the business of doing. A little bit more detail about how these channels look. So I said they're high voltage activated potassium channels. The four members split into sort of two different subfamilies really. 3.1 and 3.2 and 3.3 and 3.4 look more similar to each other, but they actually all open with very fast kinetics and deactivate rapidly as well. And the compounds, the types of modulators that we're in the business of bringing through are PAMs, positive allosteric modulators, and they increase the open channel probability of these particular potassium channels. And that's predominantly then what we see as being necessary to restore high frequency firing in neuronal circuits, particularly inhibitory circuits. So across the top, I have some examples of the way we would actually characterize this. These are wholesale patch clamp recordings for one of our clinical compounds, more potent compounds, AUT201. We can do two things really in terms of looking at how these channels work. We can pick a voltage such as -40 millivolts, say, right, how much current is there through the Kv3 channels at that point in the cell? We can add the PAM and we can characterize the fold increase, the percentage increase. And you'll see if you look at, say, the -40, then the 201, we can see quite a large fold increase is possible with a PAM. So we can drive that increase in Kv3 current that we think is useful. We can also look at the shift in this curve. So the blue shows the conductance voltage relationship in the presence of the compound compared to the wild type control conditions. And you can see in that example for 200 nanomolar AUT201, there's a shift of about 11, 12 millivolts in that curve to the left. So we're seeing more channels active, and we're seeing more channels active, particularly at more hyperpolarized potentials. So these PAMs are bringing more Kv3 channels into play where we think they can be useful. And an example, we start thinking about the translation of these, the effects of these compounds from recombinant systems into native. Later, I'll show you some preclinical efficacy data as well. But just for now, we have data from a slice preparation. This is these are slices taken from the brains of rats that have been exposed to encyclidine, an NMDA receptor antagonist used to generate a model of cognitive dysfunction. Kv3 PAMs, this is something we've seen very robustly over many compounds, many studies, are pro-cognitive in this model. When we look at the firing of their albumin interneurons in animals confirmed to be suffering from cognitive dysfunction, we note there's a lower firing frequency of these neurons. And adding in one of our PAMs, we will restore that high frequency firing that is normally characteristic of those neurons. So we can show that the recombinant, say, translates to the native situation, driving inhibitory neurotransmission. We spent many years working and thinking about the way our compounds actually can modulate these channels. And I'm pleased to share with you the results of this extensive piece of work, which was published earlier this year. This is work with Oxford University and also that's Gamma-Chi and Manuel Covarrubias in Jefferson in the US as well. And this brings together many years of work, started with cytoreactive mutagenesis, chimeras, and ending in the Cryo-EM understanding of what our compounds are doing, where they're binding, how they're modifying the channel. If I start by focusing your attention on the topology diagram, the schematic in the left corner, highlighting the turret region of these channels. And in the sequence alignment, you can see that this region is longer in Kv3 channels. This is really important. This is the unique feature of the Kv3 channels and explains why our compounds are so selective for Kv3 over other ion channels. Once you understand that turret region, pictured on the right here, is so important for forming the binding site for our compound. So in the main ion channel structure, you can see one of our compounds, AUT5, shown in red, and that's highlighted in greater detail here. And the turret literally helps to encase and form the overall binding site of these modulators. They're close to the gating machinery, the pore of the channel, so no surprise. We have a lot of understanding now about how they actually help to promote the open channel stability, opening of those channels as a positive allosteric modulator. So this is something which is true now of all of our compounds. We believe they bind at the same site and have this same molecular mechanism of action. So first acronym, PME, progressive myoclonus epilepsy. So this is where we're starting to think about, right, we spent many years developing these compounds. Where can they have their greatest therapeutic benefit? How can we show that these compounds can modulate channels in disease? So progressive myoclonic epilepsy, also called MEEC, myoclonus epilepsy and ataxia due to a potassium channel mutation, which is also called PME type 7 or then EPM type 7, was identified in 2015 as a disease that originates due to a point mutation in the KCNC1 gene, which you recognize encodes Kv3.1 ion channel. So this was the first causal mutation identified in Kv3.1 that was linked to human disease. And if you're looking at the schematic, those of you will already have spotted that the mutation occurs at one of the arginines in the S4 voltage sensor. So R320H is a recurrent mutation which has been identified in tens of patients now. And no surprise that this causes a massive impact on channel function, in fact, complete loss of function. But more than that, all subjects have one copy. So they have a mutant allele and a wild type allele. All subjects effectively then have a wild type channel co-expressed with the mutant channel. And the mutant channel actually has a dominant negative effect as well. So it's not just non-functional alone. It actually, as you can see in panel D, takes down the actual wild type Kv3.1 current. So there's a very dramatic loss of Kv3 current ion channel activity in patients carrying this mutation. And this is what led us to get quite excited about the opportunity here for a precision medicine approach, because our compounds, as you've heard me say already, are able to restore that function, upregulate Kv3 channel function. So by doing that, we would expect to be able to address the symptomology of this disease. And if we can get in early enough, maybe even the progression of this disease and have a disease modifying effect as well. So how do we build confidence in the translation of this finding? Well, we need to understand the disease. Excuse me. And on this slide, I've summarized a number of the different symptoms which are experienced by patients here. So it's onset in teens. There are patients who have generalized tonic-clonic seizures. They have myoclonus, ataxia, and slurred speech. It's all to do with poor, essentially, muscle control as a result of loss of inhibitory circuitry due to this mutation. It's a very debilitating disease. People are basically non-ambulatory, in wheelchairs, and need full care assistance as they get older. So this gives us a clue to a number of things that we might want to study in preclinical models of the disease. And what we've developed with Ethan Goldberg in CHOP in the US is a transgenic model in which the mice, through knock-in procedures, carry exactly the same mutation as the human subjects. And we've been able to study, and I've got 2 slides on this, the impact of this mutation on inhibitory circuitry and also generating a phenotypic profile in terms of behavior due to this in the mice. And sort of starting with the conclusion, really, these mice completely phenocopy the human condition. So we're actually able to pick up impact on the cerebellar and parvalbumin interneuron firing here. On the left, you'll see, if you look at the red traces where we have the mutant, so H+, you see an inability of these cells to fire at a high frequency properly. And this is restored by addition of another one of our clinical compounds, AUT6, which is also called AUT206. So we can restore that inhibitory firing. And on the right, this experiment's done slightly differently. We're looking at action potential firing success rate. Again, cells are more deficient in the wild type situation without compound. They can't fire at high enough frequency. We add in the compound and we can rescue that, be it at 40 or 80 hertz, could increase the rate of action potential successful firing. So we can shift the needle on the inhibitory circuitry in slices from these animals. But more importantly, when we start thinking about the patients, can we also treat some of the symptomology of the disease as well? I appreciate there's a lot of data on this slide. There's data across 2 different compounds, a number of different endpoints. On the left, you have AUT201, another clinical compound. Here, the dotted red line to the left in the first experiment. This is a way of showing about the enhanced seizure susceptibility in these animals. They become more rapidly hyperexcitable, have generalized tonic-clonic seizures in response to repeat injection of pentylene tetrazole. And to the right, if you can follow the color scheme here, we start providing the AUT201 compound orally. In this experiment, we can actually return these animals to more like wild type levels. So that's looking at seizure susceptibility with the ataxia part of the phenotype of the symptoms and the phenotype of these animals. Again, adding in 201, these animals are deficient compared to wild type in their ability to stay on the rotarod, and we can shift that back towards normal by addition of the compound. With 206 in the middle, similar results in the PTZ model. At the bottom, we're looking at spontaneous generalized tonic-clonic seizures. Again, addition of the compound can address that, attenuate those seizures. All of these data were published in full at the American Epilepsy Society, both last year and the year before. And the work with 206 providing an extensive data set now supporting the potential for these Kv3 PAMs to treat this condition called MEEK has been published in Cell Reports earlier this year. And it may be no surprise to you that we have now gone on to take this compound, explore this mechanism in patients with this disease. Once a mutation is identified in a gene, once it's related to epilepsy, these days that gene will end up on an epilepsy gene panel. It'll suddenly be explored as a potential explanation for idiopathic epilepsy in patients who experience that first seizure. So as soon as that happens, you start to learn a lot more about other causal mutations that could occur in your gene. This takes us much more broadly now in our understanding of the impact and relevance of Kv3 channels to epilepsy and other neurodevelopmental conditions. So just picking out a few examples here, I mean, there are hundreds of mutations identified now, so many different classes of epilepsy disorders and neurodevelopmental disorders, but certainly reinforces what we already learned from MEEK. So we have developmental encephalopathies, we have people who just suffer from intellectual disability with no epilepsy as well, for example. So these are all conditions that might be relevant to the development of Kv3 PAMs. With Kv3.2, the picture is even broader, actually. So this is now on epilepsy gene panels as well. We're seeing replication of its relevance to, for example, the DEE condition I just mentioned, but it's also bringing in what are considered to be much broader epilepsy syndromes, indications as well, generalized epilepsy, focal epilepsy, and even at the bottom, relevance to autism as well. And you'll note from the schematic I put on the right, originally in S4, this is where we had the MEEK mutation, but now you'll note there are mutations that are clearly cropping up all through this protein structure, which are relevant to disease. So there are so many ways that you can negatively impact this protein and result in human disease. So interneuronopathies, I have a couple more slides. Interneuronopathies, what we're understanding then is the clear relevance of Kv3 channels to interneuron function. So Kv3 channels can drive a restoration of inhibitory drive within the brain. That means their application can become quite broad. I've already told you about MEEK, where we have a specific impact on Kv3 channels. In other diseases, quite closely related to that, Unverricht-Lundborg disease, which is another progressive myoclonic epilepsy, EPM1, we've noted that Kv3.1 channels are reduced in terms of their expression levels in the cortex. So this is an area of interest for us as well. And something I'll just dwell on in a bit more detail, because it's a much larger indication, is Dravet syndrome. So here, the actual dysfunction in interneurons is well documented, but it originates in a very different way. It comes from mutations in the sodium channel, NAV1.1. And here, what we've noticed is even more specific in that when these animals, so these are mouse models of the disease, when these animals are studied, some go on to develop generalized tonic-clonic seizures, and it's the ones that have the lower Kv3.1 levels. So where Kv3.1 is reduced, we see the incidence of seizures in Dravet syndrome. This work was originally pioneered by Alex Rotenberg in Boston. The methodology is captured at the top, but the key result is having been enticed by this relevance of Kv3.1 to the disease phenotype, He used a AUT1 compound that was in the public domain and showed that that compound, dosed once a day, could actually attenuate the generalized tonic-clonic seizures in these animals. This was independent of Autifony Therapeutics, provided the first convincing evidence that Dravet syndrome could be treated with Kv3.1 PAMs. We were interested to replicate that study, so we brought in an AUT63 compound, another Autifony clinical compound, and replicated the findings alongside that with AUT1. So you can see here both compounds clearly shown in the same paradigm to attenuate the generalized tonic-clonic seizures in these animals. So that was a really important confirmation study for us, and now supports us going forward, looking for ways to take these compounds into Dravet syndrome as well. On my final slide, this just shows you how broad this impact on interneuronopathies could be in terms of disease relevance. So it just keeps cropping up. Some of these studies are driven by us and our collaborators. Many are completely independent, whether it's traumatic brain injury, Fragile X syndrome, schizophrenia, and more recently, Alzheimer's disease, where there's a clear understanding that hyperexcitability, comorbid epilepsy early in the disease actually drives the neurodegenerative process. This just spells many opportunities where these compounds could impact disease. So to finish, I would like to thank all at Autifony who've helped to generate these data sets, and many of our collaborators around the world as well have enabled us to put these Kv3.1 PAMs in such a strong position for therapeutic development. Thank you for listening, and I'd be happy to take any of your questions.
Thank you, Martin, for this very fascinating and very broad picture of how you can develop a drug for neurological diseases. So are there any questions here from the audience?
Very impressive talk, and I'm intrigued by your finding and identification of this very specific region of Kv3.1, because that's a long-standing problem with K channels to find specific one.
Yes.
Now, I was interested to see that you are talking about a dominant negative effect. And how do you envision that the modulator affects then and rescues channel activity? What's the stoichiometry, in other words, that you expect? And do you think that you are recruiting purely wild-type heteromeres, or do you think that you can also activate channels that are having a stoichiometry of 1 mutant,
Yes.
1 and 3 healthy 1?
Yeah, no, that's a very insightful question. Thank you for that. So yeah, we have considered that. There is a potential when we at least have the heteromer for the overall current to be increased by co-application of the PAM. So we do think there is a potential impact on channels that contain at least, if you like, 1 mutant channel. We've not 1 mutant subunit rather. We've not got into the detail of whether that's a 1 to 3, a 2 to 2, a 3 to 1, whether it works in all situations. We haven't had the chance to explore that. But certainly, I hope you understood that when I showed you that there was a residual current, a large proportion of that will be Kv3.1. But Kv3.2 is there as well. And I know I said quite a few times Kv3.1 PAMs, but the molecules are potentiating Kv3.1 and Kv3.2 predominantly. And therefore, there are, definitely residual wild-type channels that can be recruited to at least restore some of that lost current, restore enough that it can have an impact on the disease.
Thanks, great answer. Do we have some online questions? I would have one question.
Yeah.
Interestingly, you showed that some of the mutations actually affect the voltage sensor in the Kv channel, and I was just wondering how your compound can restore actually a voltage sensor defective voltage activated potassium channel.
Yeah, I think it's a related consideration. I think it is a tough challenge. We can only do the experiments and measure whether there's any, say, positive impact. And when you start thinking about the whole scope, as in collection of mutations that are disease related, this is a challenge that's coming up for many different targets now. I mean, there are over 300 genes linked to epilepsy. You need to not just understand that your particular channel is relevant to the disease, when you try to treat it, you need to understand that you can modify successfully the particular mutant and its phenotype as well. So if the data support a positive impact on even a very largely impaired channel, then that's encouraging to take that forward. But I think we need to do this in a sort of bespoke fashion using higher throughput technologies to really generate data across the whole range of mutations to show that we can have the widest possible effect. And it may be there are certain situations where we don't expect a benefit, or it's even would be contraindicated because we're mainly looking at loss of function mutations here and trying to restore, but in some situations it may be gain of function, and it may be that you want